Hi, I'm Mr. Williamson, and today we're going to talk about the limits of cell size. Why are cells so small? Probably a good idea to take some notes. I will tell you when we get to the really important, this is kind of what you need to know about cell size, but you might want to take some background information in the meantime. Again, like I've said in the past, I think that Cornell style notes are real good for most students. You might want to take it in that if at all possible. So if you're ready, let's get started. So the question is, most cells are small. I mean, small, like really small, 2 to 200 micrometers. That's 1/1,000,000th of a meter. You are made out of about 37.2 trillion cells. That's 37.2 with 11 zeros after it. Why are they so small? Why can't we just be made out of one giant cell? Okay, why do we have to have 37 trillion cells to stay alive? And it has to do with the problem of diffusion. Diffusion is the limiting factor in cell size. Again, real quick, you should know this. Diffusion is the movement of a substance from a high concentration to a low concentration. Okay? The longer the distance, the longer time to diffuse. So the problem is, if you had a cell that was, say, 15 centimeters, which is about this, okay? It's about 15 centimeters, and you are trying to get nutrients into the center of the cell, it's going to take you months to get it to diffuse that far, right? The cell needs the nutrients. It needs the resources that it's bringing in immediately. So you need to be able to get those things in. Cells don't have mouths. They don't have things like that. We will talk about endocytosis, but that's not going to cover it all. We're talking about moving oxygen and moving water, glucose, things like that. How do you get those resources from outside the cell, inside the cell, all the way into the inside? Because all parts of the cell need those resources. Again, the smaller you are, the less time it takes to do that. So here you see the surface to volume ratio limits size, okay? It's what the problem is. And what we're looking at here is if we start with a 1 millimeter cube. Again, 1 millimeter is just one of these little lines right here. All right. Sorry. 1 millimeter cube. And we just do some simple math. I don't expect you to do this math, but I expect you to understand how I got there. If you don't understand, make sure you ask questions in Edpuzzle or in the classroom. So if I start with a 1 millimeter cube, all right, simple math. The surface area is height times width times 6 because the cube has 6 sides. So 1 squared is 1 times 6. We would have 6 millimeters squared in surface area, just the outside edge parts, okay? The volume is determined by height times width times length. So it's height times width times length, all right? That 1 times 1 times 1 is 1 cubic millimeter. That's pretty much the definition. Now, if we double that size, so first of all, let's go back. If we have a cell that is 1 millimeter roughly in each direction, it would have a surface area of 6 millimeters squared, but it would have a volume of only 1 cubic millimeter. If we double that to where it is now 2 millimeters in size, again, doing the math, 2 times 2 is 4, times 6, we would have 24 square millimeters. Doing the math to find the volume, 2 times 2 is 4, times 2 is 8. So the volume would be 8 cubic centimeters. If I double the size, so if I take the size, 2 times the size, the surface area goes up 4 times, all right, from 6 to 24, all right? But the volume goes up 8 times. All right, so we have less surface area per volume to get things in. All right, so think about like if you had, you're going to feed one person? Well, they have one mouth, and so you just have to put enough food in there. This is going to be a really strange example, but you have one mouth, you put one meal in, you've now fed that one person. But now we're going to double it. We have twice the people, but we still only have one mouth. Just pretend. Or maybe we only have 1.5 mouths. So we have twice the people, but we have less mouths. So now we could only give the two people 1.5 meals. Not going to get the same nutrients. You can see the surface to area ratio, surface to volume ratio. Ratio is a comparison between two numbers. When you're at 1 to 1, 1 millimeter, you're at 6 to 1. There's 6 square millimeters to every 1 cubic millimeter. But over here, you have 3 square millimeters for every 1 cubic volume millimeter. So your surface area goes down compared to the volume. And then when we get over to the 4 millimeter, we double it again, we can just figure it out. The surface area goes up by 4 times, 96 millimeters squared, but the volume goes up 8 times. Goes up again. So now there's even less surface area to volume ratio. The surface area, we're trying to fill in, that have the things diffuse all the way in, where it can't because there's not enough surface area. This is a picture that I use in class when we are doing a lab. It's cell size lab. And what I've done is I've taken a potato and I have cut it into cubes fairly well. All right, so you can see the cube here. This one is about 2 centimeters. All right, this one is about 1 centimeter cubed. And this one is about 1/2 centimeter or 5 millimeters. All right, so you cut a cube of potato, it's just from a potato, into 3 different size cubes, a 2, a 1, and a 1/2. And then we put it into iodine. Iodine, if you remember the gizmo where we looked at testing nutrients, iodine is part of Lugel's solution. Iodine turns starch purple or blue. So when we put it into the iodine for a certain amount of time, I think it's like 15 minutes or something like that, you can see that this iodine stains the potato because it has starch. When we cut them open, we can see that the iodine has diffused into the potato. So it's probably hard to see, but it's not just the edge right here. It actually diffused in about that far. So there's a cube inside this other cube. All of the outside of this cube is stained. So the iodine actually diffused through the potato in that area. If we look at the 1 cubic centimeter cube, same thing. It's the same size. In 15 minutes, the iodine only diffuses a certain amount. It goes the same speed no matter how big the potato is. But the problem is, it just doesn't get all the way through. So if we get down to the 1/2 centimeter, it actually got all the way through. So a 1/2 centimeter, the starch diffused all the way through the cube from all sides. So the center of the cube got starch, got the iodine, and was stained. Where this one, did none, this one none. So if we're talking about resources that a cell actually needs, this area is dead. This area would die because it doesn't get the resources it needs, because diffusion takes time and there's not enough surface area to the volume ratio to get it all the way through. So as I just explained, volume increases more rapidly than the surface area. As the cell size doubles, it's 8 times more volume, 8 times more volume, but only 4 times the surface area. So every time you double the size of the cell, you have less surface area to get the nutrients and the resources that the cell needs into that cell. All right. Again, going back, just showing you this. What are we looking at? We're looking again, if you have a 1 millimeter cube, you have a surface area ratio of 6 to 1. If we double it, it's 3 to 1. We double it again, it's only 1.5 to 1. And if we went down to doubled it one more time and had an 8 millimeter cube, you would have a 0.75 surface area for every 1 cubic millimeter. You'd have less than 1 square millimeter for every cubic millimeter. And that's not efficient. You can't get the materials to diffuse through that quick. All right? So if too big is a problem, you can't make cells too big, what do? We divide them. We keep them small. If you have small cells, then the resources can diffuse through. You can have the oxygen come from the blood and diffuse in. You can have the glucose. You can get rid of the things, the carbon dioxide and the waste products from those cells into the blood. In our case, maybe it's not an animal, maybe it's a plant. Same thing. Diffusion, the rate of diffusion is the problem with cell size. You need a certain amount of surface area to volume ratio, and I don't know what it is, and it's not important. But the limiting factor in cell size is that surface area to volume ratio. It has to be fairly big. It can't be 0.75 millimeters for every cubic millimeter. It's got to be much, much bigger than that. All right? So hopefully you understand that. If you have any questions, make sure you ask them in Edpuzzle or in the classroom. I hope you took good notes. If you don't understand something, let me know. Good luck.